all. This is Dr. Mobeen Sayed from drbeen.com. Welcome to one more show. So as promised, I'm back here. We're going to talk about the serology. I have done 4 videos in the past, almost every one of them more than 30 minutes to discuss these things in details. However, there are certain interesting things that EBV does and produces this serology that we use to figure out EBV primary infection, latent stage, chronic state, or reactivation. So I'm going to go over those with you. My request once again, keep an eye on the EBV reactivation because that may be contributing to some of the symptoms that we may be attributing to spike proteins. Okay, so with this, let's start. So these are gifts from humanity. They're continuing. And this was an older lecture, so this is why the drawing style is a little old as well. This is how I started, and nowadays my drawing style is very different. Okay, so first of all, quickly, the virus itself. The virus is an enveloped virus. It's a herpesvirus. The enveloped virus, I was giving this example to someone, actually to the patrons yesterday, the folks who support this channel. So imagine, so this is SARS-CoV-2, but EBV is similar. So imagine this is a ball with some spikes on it. In case of EBV, we call them envelope proteins, and this ball is called envelope. Then imagine inside there is one more ball. Remember, like in football, there is a leather ball outside, and then there is a bladder inside that is sticking to the walls of the leather, which you inflate. So imagine that there is another one inside, and that is called the tegument, and the matrix is present inside there. Then imagine right in the center, there's a tiny ball. That ball, the tiny ball in the center, has DNA in it. And of course, this virus has many enzymes. It has these surface proteins, etc. The first interesting thing, most of the viruses have hard proteins around their genetic material. Just like we have skull around our brain to protect the genetic material that we have. Viruses have the genetic material that protects them as well. So that is called usually the nucleocapsid. So here in this diagram, this little part that is surrounding and encapsulating the double-stranded DNA is called the nucleocapsid. Then outside the nucleocapsid, and thank you very much. Thank you. So my wife just came in. We'll have, I suspect that the fast breaking time will become part of this lecture. So I'll just break fast when the time comes. Okay, so the nucleocapsid is here. DNA is inside. Outside is the envelope. Then this blue thing is the matrix and the tegument. Now, when this virus starts or is actively increasing in number, then it has some enzymes in that are working. These enzymes are like workers. For example, when we are building a house, before we can move in the house, there are workers who are building the house and they're putting things together. Similarly, when this virus is being built, there are working enzymes that are building it. Those enzymes, I made this little diagram here yesterday. So imagine this is our cell. And this virus actually lives or is built inside our nuclei. So this is our nucleus. Inside the nucleus, there are viruses that are being built. And so the workers are inside the nucleus as well. Sometimes what happens is some of those enzymes of the virus, they spill out in the cytoplasm of the cell as well. So it is possible that some enzyme of the virus are found in the cytoplasm and in the DNA or the nucleus. So can I say that these are diffused throughout the cell? They're present everywhere. So such enzymes of the virus are called early antigen D, EAD. The reason that this is called EAD, early antigen, means that these worker enzymes, they would spill out of this cell. Remember that this cell is infected, so it is going to be presenting that, hey, I am infected, to the other cells, and the other immune cells will come and kill this cell. Sometimes these cells would just cause apoptosis and die. These are all B cells, remember. The end result is that as the virus is being built, these workers will spill out even in our tissues and from there in our blood as well. So you will be able to detect antibodies against these enzymes or these antigens. And so we call them early antigens because we may not be able to see other antibodies and antigens yet, but we would start developing reaction to these workers, to these enzymes. So early antigen diffused, its presence is a sign of an active virus that is replicating. Of course, if this virus was not replicating, then there will be no such early antigen enzymes. There will be no workers. If the virus is sleeping dormant, it is just lying down. It doesn't lie down, but go with me for a second. If it is just sitting with its genetic material and just dormant or latent, then there'll be no workers. There'll be nothing like that. So we would not see that. So early antigen D's presence is a sign of active infection. So that is one. Second, this virus is such a crazy virus that, as you saw, that it can cause a B cell in which it lives. It can cause a B cell to become immortal, right? By producing reactive oxygen species that cause this B cell to become long living. In addition to that, this virus does another thing. So when it is present in a B cell, it produces enzymes that work on the B cell's DNA, and they change the DNA's expression of the B cell in a way that they convert this B cell into a blast cell. So now this is a very important thing to consider. Blast cell versus a B cell. So imagine children who are still studying in the school, they are youngsters, they haven't made up their mind for what they will become. And as they grow and they become teenagers and then adults, and then they start thinking about the world around them, and they say, well, I'm going to be a doctor or an engineer or a banker or a whatever they would like to do. There are a million things they can do. In the early age, they are not committed to some profession. But then at some point, they will become committed to some profession and they'll say, I will do this. This is my function. Same way, the B cells and T cells, immune cells, have stem cells or the parent cells that give rise to other cells. And those other daughter cells would slowly start becoming committed. Some cells will become B cells, some will become T cells, some will become neutrophils, and so on. Now, the B cell parent, that is a committed parent that would only make B cells, will be called a blast cell. So those children that are in the early stage, who have not become anything yet, who have not decided anything, you can call them by profession, they're blast children. They are not committed. They can give rise to anything. They can become whatever they want. So B cell, blast cell are kind of parent cells. And B cell committed cells are those that have decided I'm going to be a B cell and that's my function. This virus enters a B cell, converts it from a committed cell back towards a blast cell. It makes it a parent again. Now, this B cell who has become a parent doesn't know what its life is. Although it was committed, all of a sudden it has forgotten that I'm a doctor. That B cell starts producing strange antibodies. Those antibodies are really not targeted against anything, they're just antibodies. And it is so funny that this scary that this virus does it, that it converts the B cell into a blast cell, and this blast cell doesn't know what am I going to be doing, so it just starts producing some random antibodies. These antibodies can actually interestingly attack sheep cells or other animal cells. We call them heterophile antibodies. Now, there is a delicate point I want to make over here, which I hope that you will catch it. A B cell that is infected, or a B cell, general B cell, a B cell will make antibodies against a target. For example, if there is a B cell that is against SARS-CoV-2, it will make antibodies that will attack the SARS-CoV-2 spike protein or nucleocapsid or whatever. A B cell that is active against EBV will make antibodies that would attack various parts of the EBV. These are the antibodies that we would talk about shortly. But a B cell that is infected and its brain, the DNA, the brain has become mal-structured and it is malfunctioning, that B cell is speaking gibberish. It is making these antibodies not against anything. It's just speaking gibberish. It is disoriented cell. It is blast cell. It is just making up stuff and releasing it. So you can imagine the poor little B cell is all that tiny little imagine under the microscope, it is just saying, where am I? And am I all disoriented? And it is trying to speak things. And these are all incorrect things it is speaking. These are called heterophile antibodies. Hetero means someone else. Phile means loving. So heterophile, loving something else. So these antibodies love sheep cells and other cells, but not our cells or the EBV. So then, if we find heterophile antibodies in our blood, what would that mean? That means we have active infection going on, and the virus has disoriented the B cell, and the B cell is just producing gibberish antibodies. These are bad B cells. These are sick B cells. These are disoriented B cells. Monospot test actually detects heterophile antibodies. Now, heterophile antibodies aren't very good indicator of active infection because sometimes they are not found. But generally, if I said I have done serology on someone and they have heterophile antibodies, your answer will be, well, that means they have actively infected B cells that are producing these antibodies. If I said I have a patient who has EAD, early antigen diffused antibodies, you would say, well, that means there is an active infection going on and the viruses are being built, and these are the enzymes, the workers that were making the virus, and these are antibodies against them. Now, next part. Denise says buffering. I hope that my relay is okay. Yeah, monospot is not thorough. Correct. So I hope that you can hear me and you can see me and you can see the screen. Oh, Eric is here today. Hey, Eric, how are you? Long time no see. Okay, so I hope that you can hear me, and let me continue. Now, this is another important concept that I've been discussing for the last 2-3 years. So please note, this is also important to understand the serology. That is this. Imagine this is a blast B cell. We just talked about it, right? It's a blast cell. It is the parent cell. It is committed sufficiently to make B cells and nothing else, but it still is a parent cell. So this is a blast cell. Then this blast cell will produce daughters. These daughters will be called naive B cells, because they really do not know, these daughters, that what thing we will connect with. excuse me, the naive B cells, when they're produced, they produce naturally immunoglobulin D. And normally this immunoglobulin D sits on their surface and acts as a receptor for them. So the way I remember the classes of immunoglobulins that these cells make is this way. I remember MD Gia. So imagine that there is a doctor, her name is Gia, and she works somewhere and her name is MD Gia. This is the sequence of production of antibodies. So the very first time when a naive B cell comes into existence, it is able to make IgD. When it becomes bound to some antigen, when it finds its target, when it finds its enemy, then it also starts producing IgM. I remember IgM as many. And the reason for that is that IgM is actually, of course, it has two binding sites. Every immunoglobulin has two binding sites, but 5 IgM antibodies, they join together, and so they have 10 binding sites in this one group. Because there are 5 antibodies, every antibody has 2 binding sites, total 10 binding sites. So you can think about it that when we get infected by anything, we would really like to quickly mop up that offending agent. So we send in big vacuums. What are these big vacuums? These are groups of IgMs whose job is to go and just bind with the viruses or bacteria or fungus or whatever. So IgM is produced in addition to IgD first. So that is immediate response. So that means if I said to you that there is a patient, I've done their serology for EBV, and there is IgM EBV. So let's say virus capsid. Remember the nucleocapsid? Viral capsid IgM is present. Your answer will become that this means there is an acute infection, recent infection, now infection. Why? Because our B cells, when they encounter something for the first time, they produce IgMs. Then these B cells would switch. So 3 things now. If you see heterophile, that means active infection. If you see antibodies against EAD, so let's say IgM or IgG, whichever. If there is EAD being produced, active infection. If it is IgM, then new infection. If it is IgG, then previous infection that has become reactivated. So just remember, you see M, that means happening now. You see G, that means it had happened in the past. G for memory. Our memory B cells produce IgGs. They do not produce IgMs. Okay, so viral capsid IgM will be produced, and that will mean acute infection as well. Then what happens? Then what happens is that as we continue to make more daughters of the B cells, this is the same thing with SARS-CoV-2 as well. These daughters would do class switching. And we've discussed class switching in the past. Class means this, the antibody, if I make a big antibody over here, antibody has many parts. There are binding regions here with which it binds with the offending agent. And then this remaining part is called constant part. These are called variable parts. So the constant part is called the class. This constant protein has different structures. And these structures are called different classes. For example, IgD is a D class. Then IgM is a different class. Then IgG is a different class. So that is really talking about the structure of this part. When we say class switching, a B cell that was making IgM now changes its nucleus, changes its DNA's expression, splices out, meaning cuts away the possibility of making IgM, that DNA piece. It just throws it away And now the next piece of DNA is to make IgG. So this B cell, daughter cell, would now start making IgG. This IgG, it can continue to make for months and years. The same B cells would become memory cells as well. So when memory cell will become active again because they saw the infection again, they will make IgGs. So if I said to you that there is an EBV serology done on a patient and the antibodies, whatever antibody is, the type is IgG, your answer will be that this is a past infection. I haven't given you enough information to say it is active or not, but I've just said it is an IgG type antibody that we found. And you say, well, that was a past infection. Then some of these daughters would convert into making IgE. Some of them will make IgA. And that is really our body trying to protect multiple fronts of our tissues. So with this little background, if I now go back here to serology, let's look at the serology together. So it looks like a complicated graph, but it really is very simple. First of all, look at this green. This is heterophile IgM. Look at IgM and heterophile. What does that mean? Is this patient having a new acute infection, maybe mononucleosis starting, mono starting, or is this reactivation? Joseph says, good to know what helps to clear this virus. So Joseph, I just did a lecture just before this one, where we discussed what are the possibilities. For example, reactive oxygen species scavengers, valcyclovir, jancyclovir, spironolactone, and a bunch of other drugs. Vitamin C, glutathione reduced, and then NAC. So Paul Borg says new. Absolutely correct. Heterophile IgM. So number one, IgM should just give you this idea. IgM, new thing. Any infection, IgM antibody, new. Whatever kind of antibody, but if it is IgM, it is new. It is acute. So heterophile IgM, okay, good. Heterophile would give you an additional information that this virus is causing B cells to become mad. And they're speaking gibberish, and that is why they're making heterophile IgMs. Heterophile IgMs will be picked up by monospot as well. Then let's see this VC-IgM, viral capsid. Capsid is this little nucleocapsid, right? The skull of the virus. People are going to kill me for saying this is the skull of the virus, but it is the skull of the virus. So the viral capsid IgM. What is this? Again, IgM, new infection, viral capsid, viruses, capsid or skulls being made, the nucleocapsid is being made, and that is leaking out from the cells, and that is being attacked by our B cells, and that is VC-IgM. Then VC-IgG. So now we have thrown a wrench in this situation that we have an IgG over here. And I just said that when you see IgG, you say past. But here is the thing, EBV and SARS-CoV-2, both, are such intense viruses that they cause so much proliferation of the B cells, make them divide so much and proliferate so much and differentiate so much that the class switching occurs very rapidly. And so IgGs that would normally be produced within 2, 3, 4 weeks are produced right away. SARS-CoV-2 has a very similar thing. It will produce IgMs right away, and it will produce IgG right away. This IgG production is right away, but this will continue. So then how do you tell if it is acute or not? Well, if you are seeing a mixture of IgMs, then forget about IgG. This is acute infection. This is happening now. Presence of IgM is now. And why is this IgG over there? Well, our body became infected. It said to B cells that, hey guys, make sure that you beat this virus and then keep it at bay forever. So IgGs are being now formed. IgGs would go through placenta to the baby as well to protect the baby too. IgA will be formed that will protect the salivary or the secretions, including milk. IgE will be formed that will prime the mast cells to go berserk on us. Ideally, they think the mast cells that they're really helping us, but they really try to kill us. So the IgG starts forming. So good, 2 weeks, 4 weeks. Then what happens is, check this out. If I bring you here, we do not know it is 1 year past or 2 years past or whatever. If I just said, I did serology of a patient and I see VC, viral capsid, IgGs, what will be your answer? Acute infection, past infection, reactivation, what is it? Greg says, are IgG produced in the COVID vaccine? Yes. So what do you think? If I just said IgG, only IgG, no IgM, absolutely. Paul said past infection, absolutely. If there is no IgM, heterophile or VC, whatever, but no M means, and just G means past infection. So MS Amanda says, would the EBV resurgence make MCAS worse? Any infections presence and production of IgE can potentially prime mast cells. And then if we have a genetic predisposition towards mast cell being upset all the time, then we'll develop more allergies. So Dr. Iron Mom says pass. So absolutely pass. So now let's go to the next one. To explain this one, first let me show you something interesting. This naughty virus not only makes the B cells immortal, it gives them life forever. It becomes a god. And some levity, don't become upset. Then secondly, it makes a B cell go disoriented and they make gibberish heterophile antibodies. It does something more as well. It becomes latent. It lives in us forever. 90%, 95% of adults have it in them forever. So how does it sit in us forever? It sits down in the B cells. It has its genetic material. The remaining virus goes away. The envelopes and the virus proteins and the enzymes and all those things go away. Just the genetic material plasmid becomes present in our nucleus of the B cell. Now, it doesn't integrate there. It just is present there. There, it only opens up a few genes, not all of its genes, because if all of its genes are working, then all kinds of worker enzymes will be produced. They will start making new viruses. It only opens up LMP genes and or eBNA genes, some genes. These genes, these genes, have you heard sometimes that the pregnant mothers, their babies send fetal cells, their stem cells, to repair the mother and to help her tissues if she becomes sick while she's pregnant? It is the same situation over here. These genes are producing little enzymes that are keeping this B cell immortal, that are asking this B cell to continue to divide so that this genetic material continues to divide as well. So Denise says that, did you see they found mast cell degranulation? Chemokines interact with nervous system receptors to cause the plummeting of body temperature for anaphylaxis and likely also sepsis. Whoa, I did not see this. Denise, if you are in Discord, can you send me the link? I would love to see it. Yes, and Joseph, please watch the previous lecture as well. Thank you very much for that. So back here. So this little genetic material is producing enough enzymes and enough workers that are asking this B cell to continue to divide and stay immortal. This B cell is not going to die. Just like Bruce Patterson found, Dr. Bruce Patterson and his team found that the spike protein S1 is sitting in monocyte and the monocyte becomes immortal forever. Same way, this B cell is now immortal. So this eBNA is being produced. That means we will have eBNA antibodies being produced all the time as well. So if it is a virus that is sleeping in a latent phase, medical students, when they hear from me sleeping, they say viruses don't sleep. So correct, latent phase. So this is a virus in the latent phase. I do see this virus sometimes under the microscope or electron microscope, having its pillow and lying down. So I have that idea. So anyways, latent phase. What will you see in the latent phase? Number one, you will see IgG, which is an indicator that somewhere in the past the virus has arrived in our body. If the virus never arrived in our body, then there'll be no IgG and there will be no other protein. So for serology to be positive, there is going to be IgG. That just tells that we were infected. That's a fingerprint of previous infection. Then if you see eBNA, that means virus is now sleeping. It's dormant, it is latent, and it is just keeping the host B cell active, dividing, and immortal. So if I said to you that I have a serology where I see viral capsid IgG plus I see eBNA1 IgG, what would you say? You would say that this is latent phase. Latent phase. In the latent phase, normally EBV doesn't do anything to us. But the problem is from this latent phase, it can become reactivated. So my previous lecture today, emotional stress, immune stress, virus stress, reactive oxygen species productions, all of those things can reactivate it. So think about it before I go to the serology for reactivation. If the virus becomes reactivated, not reactive, and if it is producing new viruses, don't you think that the workers will come back? Don't you think that then some heterophile antibodies might be produced? Sky Frog says, I'll be latent after melatonin tonight. Yes, you will be. Right? So this was a latent virus, sleeping virus. And then it woke up and its genetic material became active and it started saying, well, let me make more viruses. So now it is going to make more viruses. If it is going to make more viruses, isn't this true that heterophile antibodies can come back? Monospot test may become positive. It's not always possible. But more importantly, EA, early antigen, the workers that build the virus, the diffused ones, will start seeing antibodies against them. That means the virus is building itself. That means it has become activated. So what will be the profile? So many medical students and professionals look at this and they kind of fall flat. But this is actually very simple. This was acute, this was latent, and this is reactivation. So what do you expect in the reactivation? First, IgG has to be present. If there is no IgG and just IgMs, then that is not reactivation, that is new infection, primary infection we'll call it. You have IgG, that is a sign that it was passed. Then you have early antigen antibodies being produced. That means it is now being built again, that is it becoming active again. A past infection became active again. You may find IgMs, but you may not find IgMs. You might just find antibodies against EA. And that is sufficient to say this virus is being produced. You may find heterophile antibodies, you may not find them. If you find them, once again, with IgGs, no IgMs, we have reactivation. So now when the reactivation is occurring, this is very important that eBNA, that little system, excuse me, let me just quickly break fast. The eBNA, that was the alarm clock of the virus, that it was sleeping and saying, all right, eBNA, you just keep the B cell active. That eBNA would go away. The virus doesn't need that worker anymore. It needs the new building workers. So eBNA would go down, EA would come up, IgG would be present. This will become reactivation. Done. It's simple. Now, in addition to this is the best doctor clue now. If you suspect that there is EBV, but you don't see those in the serology yet, look for the downy cells, look for the active T cells. You might actually find downy cells before you find all the IgGs. So that means long COVID patient or vaccine injured patients, or even ME/CFS patients that are actually suffering because of EBV reactivation. You might do serology. So monospot, one, not very reliable, but still serology, and then T cells. There can be more. You can do DNA from the saliva. And as I said in the last lecture, there are actually possibilities to do DNA antibody, saliva antibody fingerprints, but these are not generally available. So this is the discussion. What did you think? Did you like it? Jenny says, can you be infected with more than one variant of EBV? Does this have a bad effect? Yikes, never thought about that. Glad I'm germaphobe already and not in the orgy culture of the day. So, number one, it's going to be a really difficult thing for us to not have EBV. The very first kisses and hugs, not hugs, just the kisses, and the interactions with others will cause EBV to become part of us. Now, I don't think that we have simultaneous production of various strains of it. So I think you should be okay there. So, Dr. Joey, very good question. Would this apply for patients that had EBV 20, 30 years ago and had COVID? Correct. EBV, mostly the trend in healthy individuals is somewhere in the school time when people start kissing each other, the children start kissing each other, that is when they get EBV. Some of them would develop mono, some would not even develop mono, but have EBV. That EBV will become latent in them. Now, 50, 60 years later, they might have a weaker immune system or immunocompromised for some reason, or drugs, or transplants, or SARS-CoV-2 like infections, and the EBV will reactivate. That is the point of the whole discussions today. That EBV is reactivating even in the older people, and the reactivation in acute cases 55%, in the hospitalized patients 79 to 85%, and in long COVID 66%. So good question. M. Gregory says sleep calms immune system. Yes, sleep actually does a lot of benefit to us, including improving our immune system's behavior. Correct. So this is a very important one, that EBV latent in various stages can actually be associated with various cancers as well. Because it is just continuously bothering the immune B cells and asking them to stay mortal, sometimes these B cells become really immortal and start becoming cancers. Joseph says, my doctor says there is a difference between mono and EBV. Okay, so I think here is what he may mean. I cannot really know what he was saying. Mono is active infection of the EBV, and then EBV is usually used as latent EBV sitting in the body. So he might mean that active versus not active or latent. That's okay. But generally, EBV is EBV, mono is caused by the EBV, and so on. Par says, I'm watching Dr. Bean renal physiology and texting in live chat. Very good. Excellent. I have to do some more lectures on renal as well. Boone says, this has been a great explanation. Thank you. You're very welcome. You can also do me a favor and like, subscribe, and share. So Don says, is there a reason for IgM to continue to rise over a long period, 1 year plus? If for some reason the B cell become bad, I don't want to say cancerous, but tumorous, for example, and they just keep making more B cells and these B cells just keep making IgMs, that is possible. This will be difficult for a infection to just keep making IgMs. That's not going to happen. This is also going to be difficult that every 2-3 weeks there is a new kind of infection that produces a new kind of IgM. So this seems to be an abnormality of a B cell and should be worked up. Neepa Gandhi says, can we do a comparison of methylene blue with vitamin C? Yes, we can. Will do. EBV leading to hypersomnia? I have not heard that. Maybe, but I have not heard it. So old school says, is ovarian cancer one of those cancers? So let me actually open up that list of associations and go over that together. Let me just make sure that is the right paper. Okay, so I'm going to share my screen to go over the list of cancer. So this is the last lecture, actually this lecture as well. The top first link has this. So first I want to go here. The latency of the virus, meaning it is dormant, it is not doing anything, is of multiple types. Type 0 latency means there is no EBV gene at all, and possibly there is no EBV at all. Type 1 latency is what is the eBNA being produced, but nothing else. That is the alarm clock and keeping the cells dividing. Type 2 latency is that some more genes are working, eBNA1 and LMP1 and 2. Type 3 is actually a full-blown virus that is building itself, and that is these various genes here. Now, with this, type 1 latency, that is eBNA only, is associated with Burkitt lymphoma and can be modeled in Akatomutu. So that is one. Type 2 latency can be found in nasopharyngeal carcinoma, Hodgkin lymphoma, and T-cell lymphoma. Type 3 latency is seen in lymphoblastoid cell lines. So these are the kind of cancers that are associated with EBV. Old school says, thank you, you're very welcome. So with this, Skyfrog, don't take MB drop directly on tongue. It will stay blue for days. Ask me how I know. So I'll tell you a funny story. Someone reached out to Dr. Paul Marek and said that here we make MB troches. Troches are pills that you can keep under the gum, and or between the gum and the cheek, and they just keep dissolving there. And so Dr. Marek, actually, he's a very brave physician. So he actually tested it. So he put the troche in between the cheek and gum. And then he sent a photo because his whole mouth was blue and he has his mouth open and said, look, I'm all blue. And the troche stayed there for 20 minutes and his mouth stayed blue for the whole day. So that was funny. MEBV, EBV. So M. Gregory, ideally, intermittent fasting allows the autophagy to occur, which could cause EBV to become cleared as well. But I have not seen studies yet. And that doesn't mean there are no studies. I just haven't researched it. It is possible. Jeremy says, I had mono in the summer 2003 and then bucket. I'm sorry about that. So with this, thank you very much. Have a good day. Like, subscribe, and share. If you would like to support this work, there are links in the description. And just like other Cool Bean, if you would like to invest in Dr. Bean, you can, of course. And we'll go from there. I would see you tomorrow. Bye bye for now.